Our first speaker this morning is Dr. Josef Coresh. For those who do not know, Joe is the Terry and Mel Karmazin Professor of Population Health and Medicine at the New York University Grossman Medical School, where he is also the founding director of the Optimal Aging Institute. Within that center, their mission includes prevention and treatment of aging-related diseases. And Joe is probably one of the most internationally recognized scientists out there. Joe, I got to say this because it's almost staggering to me. So Joe's publications have over 350,000 citations. So if you look him up in Google Scholar, he's one of the most highly cited authors in the United States. He has done a massive amount of proteomic work in the cardiovascular and renal areas. He's a huge amount of work in renal as it relates to cardiovascular. And he leads the ARIC NCS Proteomics Initiative, where they have over 150 million longitudinal proteomics measurements. So a world authority on proteomics and the application of proteomics in human health. Highly cited author within the US and beyond. Absolute pleasure and an honor to be able to introduce Joe. He's not able to make it here today, he has to present virtually, but just the fact he was able to take 30 minutes out of his incredibly busy schedule to come speak with this is a real honor. So Joe, thank you so much. I don't know if you can see the room or not, but the room is absolutely packed. We've got standing room only here.
Yeah, I can see the room. I saw the end of your presentation. Can you hear me clearly?
Yes.
And you can see my slides and the little red laser pointer. Thank you for the kind introduction. I apologize for not being able to fly and be with you, but hopefully you can hear me and see the presentation. So we'll go. And thanks for the extra time. One can always use extra time. I've got a lot of slides, but I'll try and give you a whirlwind of this exciting time that we're living in. I think, I have been doing, research for over 30 years, and I remember the difficulties when we couldn't multiplex SNPs and spent a lot of times to do that. And then the arrays came in and it was just unbelievable the first time I saw those arrays. And then the first time I heard of proteomics, I wasn't sure I believed it. And it seems to be true, but as always, there's complexity. So I wanted to talk about the journey from 1,000 to 11,000 plasma proteins. And basically, the outline of the talk is to talk about discovery platforms and the complementary approaches between SomaLogic's approach, which has gone from 1,000 to 11,000 proteins. And when I heard about that last year, I was just, does it work as well as it did when it was smaller? And then Olink has grown from 1,000 to 5,000 proteins, same question. And the two we've known for a while only agree some of the time. So those of us who are consumers want to know, what does that mean in terms of science? And the answer is proteins are complicated, and I'll show you what I think that means. And then in terms of precision and accuracy, and then briefly the fit for purpose, right? We do discovery, we do biology studies, and we do specific targets. And I think the answers are a little bit different for each one, and I'll briefly touch on that. Okay, so my journey with proteomics was, I tried mass spec proteomic collaboration for epidemiology for 10 years with a world leader, and it just didn't work for me. It turned out too much pre-analytical variation. Then I learned about the SomaLogic platform in 2017, and we decided to do a pilot study to look at quality control, and we were impressed. We got reagents as a gift from SomaLogic at that point. So then we established a team, and it took us over a year to really get an NIH agreement to actually assay a huge number of samples in ERIC. So then we actually, in 2020, got proposals and got 5K proteomics on about 25,000 people in ERIC. And then within a year, we had our first paper by Keenan Walker in Nature Aging about dementia proteomics. And now we've got 170 manuscript proposals in ERIC, over 53 published papers. And this timeline about team forming then says adjourning, but you never adjourn. You always go back to the beginning in science. And really, I'll talk the most about this little study we've done recently that we're about to publish about the 11K platform and the 5K Olink platform. So this is an update of a series of different papers about proteomic comparisons. One of the most well-known one is this paper in Nature in October 2023, which had many fine features and showed what we've known for a while, which is that Olink and Soma sometimes have excellent agreement, sometimes have okay agreement, sometimes have fair agreement, and often have poor agreement, right? The thing is, the corrected version of this paper shows slightly better precision with the SOMA platform than the Olink platform. But it was an unfortunate thing that the original version of the paper and this one figure really had SOMA over here. And that was because the CVs included pairs of samples from different days, whereas the intention was to have the same day, the same sample, split duplicates, to look at just analytical variation. So the analytical variation, the precision with SOMA was excellent. This is the overlap between the 5K SOMA and the 3K Olink. The question is, what happens when both platforms have moved one step ahead? So we've been interested in this quality control question for a while now, and we developed this calibration panel with 116 participants that are balanced across risk of cardiovascular disease. Half develop cardiovascular disease within 5 years, half don't. They're balanced by age, by GFR, by sex, and by race. And we've published a series of papers. One of them is in Clinical Chemistry in 2023 by Mary Rooney about the SomaLogic 5K and Olink 3K platform and some targeted markers. And then I'll talk to you about what's posted now in a preprint server, and we're revising it for publication between the comparison of the 11K and the 5K platform. So we first always do quality control, and generally that's rather boring, and it generally worked with 115 out of the 116 participants in one run and 103 in the samples that were over 25 years old. And we got to keep about 10,000 aptamers representing about over 9,000 distinct proteins. So the SomaLogic cleaning was boring, and in some sense, the quality control was boring too. So the duplicates had a coefficient of variation of 6.8%, very similar to what we published before, and the correlation in human plasma in older adults had a median of 0.85 if you're ANML normalized, and 0.86 if you're median normalized. So in a way boring, but in a way boring is actually very exciting because it means that we can get to this coverage of over 10,000 aptamers with the vast majority having coefficient of variations, half below 7% and nearly most of them below 10%. What happened when we looked at samples that were over 20 years old? The same thing. In a way boring, but in a way, it tells us that at minus 80 degrees Celsius, proteins stay, many of them quite stable, and the measurements are quite reproducible, which is exciting for us because we've also done discovery data that I won't show you about dementia and kidney disease and diabetes with our very old samples. This is kind of what it looks like when the correlation is over low values or high values. And when we compare our visit 2 data, 25-year-old samples with 5K proteomics done several years ago to 11K done now, the correlations are between 0.79 for Spearman and 0.88 for Pearson. And that's actually, I think, quite impressive. And the 5K array sits inside the 11K array largely. This is what happens when we compare it to a targeted cystatin assay done years apart. We get a correlation of 0.9. Not every protein is like this, and I'll show you some of that, but the ones that are correlated stay stably correlated and similar on the 11K as it was on the 5K platform. So in a way, SOMA is boring, but that is great. If you can double your size and stay the same, that is great. Some assays stayed uncorrelated. Albumin correlation improved a little bit to 0.5 from 0.2 or so earlier. So somewhat better, but not a lot better. Often when things are uncorrelated, in our hands, they weren't so predictive of biology because they're totally novel. Sometimes they are, and I still don't know why, but despite being low correlation, the SOMA albumin measure is just as predictive of mortality as the clinical albumin measure. So if somebody can sort this out, we would love to collaborate, and then it would move up higher on our priority of papers to publish, because we have this observation for a couple of years now, and we don't know why. Something that Soma has measured in albumin, maybe it's an unmodified albumin, and then the modified albumin that's in total albumin is still measured by the clinical assay, but somehow you get orthogonal prediction. So what happened with our Olink 5K assay and the correlations? Basically, the quality control worked in 105 out of the 116 participants, but there was a surprise. Out of the 5,400 proteins, 2,000 had a coefficient of variation greater than 50% in our human plasma. So what happened, right? When we looked at our coefficients of variation, we had lots of proteins below 20%, but a lot with very high CVs. And the correlations, we had ones with high correlation, but a lot with low correlation. So we were just surprised, well, did we mess this up somehow? Well, if you look at a comparison to Cystatin C, based on duplicate assays, batch 1 and batch 2 are highly correlated with Olink. So at least the pairing for cystatin wasn't messed up. And since the assay is done all at once, that means the other pairing wasn't messed up. So what happened? It turns out if you make this graph, you see that some proteins are detectable above the limit of detection 100% of the time with Olink, about 1,500 proteins out of the 5,000. And then some are above 90%. What I hadn't fully appreciated, but when I reread the website, I realized is that there were many proteins that were intracellular proteins, and in plasma, they were below the limit of detection published for that particular Olink assay. And as you might expect, and as you can actually see in the corrected supplement to the Nature paper, even with the earlier assay, what happens is when the percent above the limit of detection is 100%, the coefficient of variation is closer to 10%, maybe a little bit higher. But when the most values are below the limit of detection, the percent above is closer to 0, not surprisingly, the coefficient of variation gets very high, and it can even be 100%. So what can one do with that, right? One option is with troponin in the clinical assay, what we've done with the early assays is if the value is below the limit of detection, we round it to half the limit of detection. And magically, that creates agreement so that low values are just said to be low, but we don't know how low exactly because it's imprecise at that end. And then, it seems like magic, but the coefficient of variation changes to 14%. And now we're sort of more of what we were hoping for. But there is a subtle thing here that you just kind of have to think about, understand in using the platform. And what we found is that, not surprisingly, the proteins that were in the initial 1K for Olink, for us on the overlapping with Soma, that was 600 proteins, had a median CV of 9%. And the percent above the limit of detection was 100%, right? And the Soma CV for those was 6%. As one added proteins, right, what happened was you needed to round them to lower the limit of detection because those proteins often had, instead of 100% detectable, more undetectable. And the ones that were added from 3K to 5K were mostly intracellular, and in the plasma were mostly undetectable, which means when you round them, you get to a good CV, but if you don't, you stay at the high CV. And interestingly, it's not just abundance, at least for the Soma platform, the precision stays pretty much the same. So this is a summary of what you see with all of these data in the preprint, depending on how you analyze the data. So the Soma scan 11K and 102 people had over 11,000 different protein measurements with a median correlation of 0.85, a median CV of 0.68, and it pretty much was independent of whether you're on the 5K, the 7K, or new to the 11K platform. On the right, we made a graph like the Nature paper. Only thing is, I think one should look at the number of proteins, not just percent of proteins. Because if you're doing discovery, it matters that you have 3,000 proteins here with a coefficient of variation of about 7%, and more than half of the 11,000, so 6,000 or so proteins, below 7%, right? So this is the distribution of CVs for the Soma 11K platform. For the Olink Explorer 5K, if you don't round, the distribution has a median CV of 35. The raw data, if you try and use the data below the limit of detection, you get a peak of many proteins with a CV greater than 50%. If you're not interested in low values being different than each other and just declare them to be all the same at the limit of detection over 2, that imprecision goes away, that bar becomes much smaller, and the distribution shifts towards here with a median limit of detection of 13%. And you can see that that issue is much bigger. The coefficient is 10% for the oldest platform, things that were on all 3 platforms, versus the Olink 3K when it goes up, and then the 5K, the proteins that are only new to the 5K compared to the 3K, which were 2,500 proteins, often they had poor precision, but if you round them above the limit of detection, they have a precision of 14% CV. If you exclude the values, you get pretty much the same thing, but you have a lot fewer values because you're excluding values. So this is sort of the bottom line. What happens with the correlation between the platforms? Again, it's not too different from before. Some very high correlations, some medium correlation, of low correlations between platforms goes way up. But the interesting thing is, it's if the proteins are 100% detectable by Olink, then it's like 1/3, 1/3, 1/3. If the proteins are mostly below the limit of detection in human plasma, then mostly the correlation is close to 0 between the platforms. So this is our sort of conclusion on our analysis of the comparison is that, you know, as Eric updates from 5 to 11K, precision trends diverge as Olink antibodies added are mostly to intracellular proteins. And what happens if you look at the details of that? I think looking at raw data is very helpful. It's hard to do when you have omics, but I think it's important to look at least examples and know what's happening. So when the correlation is very low, a lot of the values with Olink are below the limit of detection, but not consistently so. And the same thing happens here, not consistently so. So this is not going to get completely fixed by just rounding things to half of the limit of detection, which is 1 unit below the limit of detection on the log base 2 scale. On the other hand, when you get correlations of 0.6 or so, if you take all of these values below the limit of detection and you round them to 1 dot here, then sometimes you get disagreement, but often it's low, low. And if you don't care about those from the high, you actually get some interesting, useful data. And when the correlation's higher, you see lower precision, but not terribly lower in some values that are above that. This is when the correlation is high. And for interleukin-6, most of the values are above the limit of the
going for a while. Hopefully they're working and getting reconnected quickly. We did, while we're waiting to see if we can get reconnected, we did have a question from Zoom. I do not know the answer to this, so maybe some folks in the room do. Has there been a comparison between plasma and serum samples? Also the anticoagulants, et cetera, citrate versus EDTA. So have you done any studies at Soma kind of making those comparisons?
Joe, are you familiar? Yeah. We do comparisons, and for the most part, between, you want me to speak in the mic?
Yeah, that might not be a bad idea. Or here, we should have one.
Thanks. Yeah, we do those types of comparisons, and for the most part, the proteome correlates really well between plasma and serum. Most of the samples we run in plasma are EDTA plasma, but we've also looked at different anticoagulants as well, and the correlation between the anticoagulants within plasma are a little bit better than between plasma and serum, but for the most part, they correlate pretty well. There are proteins, though, that don't correlate well between plasma and serum. Most of that work that we do when we're thinking about that is based on the soma signal tests that I'll talk about here in a little while. Most of those were developed in plasma. Some of them were developed in serum. And for the most part, about 90% of the tests correlate really well between the two matrix, but there's a couple that don't. So I guess the short answer is generally there's good correlation, but there are specific examples where there's not, and we have that information and we can share that. We don't see too many people run a large plasma experiment and a large serum experiment and try to combine the data sets. I haven't seen that necessarily, but it likely could be enabled if we get the right information.
Do we have any word on reconnection of Joe?
Rich, we're getting him back online.
We're getting him back online? Okay, so they are getting him back up online, so that little extra time we had will likely be vaporized, but hopefully we should have him back here momentarily. While we're waiting for that, which should hopefully only be another minute or two, any other questions people might have come up with in that time that they didn't ask before? Yeah.
What about the storage time, -20 versus -80?
I'm sorry, I didn't quite.
Sample storage time.
Sample storage time?
-20, yeah, for serum.
Oh, -80. So I think what Joe was just talking about is at -80, they saw very, very strong correlation. Those samples were what, 10 to 20 years old? Something in that ballpark at -80. So at -80, very, very good correlation over pretty old samples. I don't know about -20. Some folks, do you have any knowledge about -20 storage for samples?
Yeah, we have looked at it as well, but we haven't looked at it to the extent that you just saw 5, 10, 20-year sample stored at -20. We just don't run too many samples like that. We know the samples are stable at -20 for a shorter amount of time, weeks to months. We do those types of experiments because we actually bring in samples from clinical sites and run the soma signal tests on those. So we have confirmed that shorter term storage at -20 is okay, but we haven't looked at things that are 5, 10, 20 years old.
He's back down here, but it's not up on the top screen. So we do have, there we go. Joe's back. We can see, he's muted. We can see it. Oh, there we go.
Hello.
Yeah, there.
I apologize. When did I disappear? And I'll try and do a quick preview. And I saw now that gratefully you've taken over and talked about -20 storage, which is tricky. I think -80 is amazing.
IL-6 slide, Joe.
IL-6 slide. Okay. So actually, I think from there, basically, okay, so that's not that long ago. So cystatin correlations were good. And then and correlations with other markers were good. So then I wanted to talk for a few minutes about this precision discovery versus targeted markers. You can hear me?
Yes.
Good. I guess I muted the phone, so I couldn't hear that you stopped being able to hear me. All right. And I'll put on the laser and we're back in business. So for discovery, I think I won't show you a lot of the papers. I'll show you sort of this limited sample because I think it's very illustrative of these nuances of what can happen with these platforms. But we do know that even though aptamers disagree with antibodies a lot, when you do discovery, if you replicate it, it seems to replicate and then be replicated over and over in other studies. And sometimes it's aptamers that are the same as the antibodies in a familiar assay. And sometimes I think it's really, truly novel. So what I wanted to show you was a small example where in the 116 samples, we said, well, what can we study? Well, they were half men, half women. what are the differences between men and women? And even with 116 people, you could get a p-value of less than 10 to the minus 10th for about 12 proteins on the Soma 11K and about 12 proteins on the Olink 5K. The overlap actually has more discovery in Olink than in Soma. So it is an interesting thing. Whenever you do comparisons, you should know whether you're looking at overlap or the full platform. And if you have a bigger platform, you're going to get more hits, often in the non-overlap part. So of the 12 Soma proteins with a sex p-value less than 10 to the minus 10th, 3 had a correspondingly hit on Olink, and it was strongly correlated. I'll show you that. 9 did not. Of the 11 Olink protein with a p-value of less than 10 to the minus 10, only 3 had a strongly correlated Soma hit protein. The other 7 had a weakly correlated Soma protein, and 1 had no corresponding Soma. So Soma covers almost all of the Olink platform, but often it may be an uncorrelated. So it is quite tricky in the sense that proteins may have many features, right? So this is an example of where it's fairly simple. So pregnancy zone protein is higher in women than in men, a p-value of 10 to the minus 11 on both Soma and Olink. There's a correlation, but it's curvilinear, right? And you can see that amazingly, Soma has incredible precision between 2 replicate measures, even in men with low values of pregnancy zone protein. Whereas Olink, as you might predict, at lower below limit of detection has lower precision, but really you can tell apart women from men. So this is an example where it's still quite useful despite the low precision on the low end. You know, these another protein where basically the same thing happens, another protein where basically the same thing happens. And then things like PSA, Soma has it, Olink doesn't. And amazingly, PSA levels in women, right, at very low levels are very reproducible across 2 different aptamers on Soma. Luteinizing hormone, as expected, higher in women than men, amazingly great precision in men, and presumably here women with oophorectomy. This is FSH, same thing, again, amazingly great precision at the low end, and then again for another platform. I think it gets more sort of interesting, surprising when you get to the other end, the Olink proteins, right? So now sometimes you're basically getting this Olink protein, acrosomal protein, higher in men than women, lower precision in women, but Soma, despite having 2 aptamers for this Soma assay is just not correlated with sex, right? So this happens, and it can happen again with things, other proteins, you know, even though Olink is imprecise here, for some reason, Soma is just uncorrelated and not different between men and women. This is an example that's more compelling. Epididymal secretory protein, higher in men than women, as you might expect with Olink. But Soma for this protein must grab some very different part of the protein because it's not different between men and women. So I think overall, you'll see my conclusion, and the same thing with FSH subunit beta. So I wanted to sort of conclude by saying, right, that the Soma Olink 11K platform has unprecedented coverage. Really, I think quite amazing, retaining amazing precision with a coefficient of variation of 6.8% as before. The Olink HT5K expanded the coverage, but includes many proteins with human plasma levels below limit of detection. So you should be aware of that when you're looking at a specific target, right? And below limit of detection, precision is lower, but it could be informative that it's just low and it changes the coefficient of variation depending on how you treat it. Discovery benefits from wide coverage and novel assays, but targeted proteins with assays of known biological meaning, like albumin or cystatin C, you want to know that broad protein coverage platforms may or may not correlate with the previous targeted assay, right? So SomaLogic's expansion is unbiased, it retains precision, but it hasn't improved much on the correlations to known targets. Some have improved, but staying consistent means that, at least for me, for dementia, when I want to measure, A beta or p-tau or NFL or GFAP, in plasma, Soma hasn't been as useful for these targets for me. In CSF, levels are different, and I'm told that at least one of these may be at the right concentration to do better, right? Olink, again, sometimes hits and sometimes not, but because it's antibody-based, it's likely to be correlated with another same antibody type assay. So I think discovery versus targets is different. And then sometimes with the discovery, you get a specific hit that's a novel measurement of that same protein and an aspect that you hadn't appreciated before that you need to. So thank you very much. I apologize for the technical issues, but hopefully you were able to hear me now, and we're still a little bit ahead and have room for questions.
Okay, thank you, Joe. That was a really interesting talk. I'm sure we'll probably have a couple of questions here, and yes, we do still have time. So questions in the room for Joe?
We may need somebody with a microphone for me to be able to hear you.
Yeah, we just got it.
Can you hear me, Joe?
Yes, now I can hear you.
I wanted to ask a question based on that last point, specific to thinking about the brain and identifying these markers, and how you're thinking about delineating a certain disease process versus a possible just marker of brain aging that demonstrates susceptibility of an individual to various pathological insults that might manifest in CNS diseases. Because you said there you needed to understand the biology or more specific targeted markers for, say, Alzheimer's disease. But if you look in some other dementia population, if you're identifying something by like a SomaLogic platform that may be a more general marker, how are you thinking about that stratification, or are these biomarkers of like a unified mechanism?
So excellent question. It's occupying a lot of our time. I would recommend to you, Keenan Walker has recorded a webinar about the discovery studies. Many of them have been done with the Soma platform, but he's also correlated them with blood, CSF, expression of RNA, and other platforms, and Mendelian randomization. I think my lesson from our discovery studies is that clearly there are multiple pathways involved in Alzheimer's disease. And Keenan has also shown that the balance of the pathways is different when you're in the last decade before Alzheimer's disease versus we get a signal with the Soma panel 20-some years before dementia, right? So very early, we get a signal with inflammation and proteostasis. And then later, you get, a different balance. And then importantly, very late in the disease, you actually get marked elevations of the, amyloid proteins. And in CSF, you're getting to a diagnostic stage, right? So at this stage, I think you get different pieces, and we're actually correlating what we get with the standard understood Alzheimer's type markers to the broader pathway with Soma to understand the full pathway that's correlated, right? And it's interesting also that despite not having a good measure of p-tau or amyloid, our total ability to get a dementia score is actually quite impressive. So it suggests there's redundancy in the proteome, and we can see other aspects, but it is different.
Great, thanks.
Any other questions for Joe? Well, Joe, thank you again. Really appreciate that. Was a phenomenal talk, and some really interesting data coming out. And you said this, so that was the meta-archive version that you were showing us the data from?
Yes, and right now we're revising it for publication. So hopefully within a couple months it'll be out.
Great. Thank you again so much. Really appreciate it.
My pleasure.